phenomenon of its fluid nature. In the class, we have discussed that the membranes are fluid-like structure. Today we will discuss about the experiment by which you can prove that membranes are fluid. But before that, let us look at the complete organization of the membrane. So the structure presented in yellowish color is the one which shows you the fatty acid chain. And the top of it, the bluish color, light bluish color, is the head group of the lipid. So if I select one of the lipid, I'm just selecting one of the lipid. So here you have a head group and you have tails. So these are the representation of phospholipids which we have discussed. So this is given in the green color also. This is the one which can, show you the complete structure of lipid. Then what you see is that there are certain structure. This is structure. There is a complete structure. This is the one which is a protein. Now this protein could be the one which is spanning the membrane, and they are present inside the membrane. And of course, part of it is on the outer surface of the membrane, and part of it is on the inner surface of the membrane, and they are called as an integral protein. So what you have is that it is present transmembrane. It is present across the membrane, and you can see the helical structure is moving, and part of it in the outer side of the cell, and part of it is in the inner side of the cell. So when I am explaining this, you can assume that there is a cell. So this is a cell where this is the outer side of the cell, and this is on the inner side of the cell, and this is the bilayer membrane. That is a three-dimensional structure. This is the one which shows you the bilayer of the membrane. So this is the one which is actually the membrane structure. Now this gives you the three-dimensional view of the membrane. So okay, let me just go back and yeah. So you have understood that there are certain proteins which are across the membrane, and they are the one which are integral protein. Integral protein means if I want to remove this protein, I have to break the membrane. Otherwise, they are interacting with the membrane lipid. So the portion which is present in the membrane, they are, suppose this part here, this part of the membrane protein, what should be the nature of this part of the membrane protein? They should be hydrophobic. So the one which is crossing the membrane should be hydrophobic. So you have a hydrophobic amino acid. Why you have so? Because they can interact with the lipid layer, and their interaction can be stabilized. And the one which is present on the outer surface, they may be hydrophilic. Mostly they are hydrophilic in nature. So what you have learned is that you have a membrane lipids and proteins, they are interacting together. There are certain proteins which are not present across the membrane. So they are present in the periphery. So for example, this is the protein which is present on the inner side of the membrane. So suppose I have a cell. This is the inside part of the cell, and this is the outer side of the cell. So what I'm representing is that the membrane which is taken from here, the membrane which is cut out from here, that is what has been shown in the three-dimensional structure. So you have learned about the membrane protein, you have learned about the general lipid structure. What else could be present in the lipid? The lipid you can see is that could be a glycolipid. So now this glycolipid refers to the lipid where your sugar unit is attached to the lipid. So you have a lipid. So suppose this is a phospholipid, and with this sugar unit is attached. So it becomes glycolipid. You can also have the glucose part or saccharide part attached to the protein, and this could be oligosaccharide chain of the glycoprotein. So this protein could be glycoprotein. Why glyco? Because it has got carbohydrate unit attached with it. So you have the peripheral. So if I look at all the lipids, what are the lipids? Let us talk about the lipid. So you have a phospholipid. This one is a phospholipid. This one is a glycol. Any other? There is suspended here. This color, the orange color. This color protein, sorry, this color molecule is known as sterol, and most of the animal cell, they have cholesterol. So this is the sterol. How sterol is present? Sterol is embedded in the lipid structure. You will learn about the function of the sterol. They are again a lipid molecule, and they are interacting with the, how they are interacting? They are interacting with the fatty acids present in the membrane. Now, looking at the protein, what type of protein you have? You have a two types of major protein. One is integral protein, another is a peripheral protein. Both integral protein and peripheral protein, they will be attached with the membrane, this part of the membrane, by, no, uh, in most of the cases, you will find that they are attached by non-covalent assembly, but some of the peripheral protein, they can also be attached by a covalent linkage. So here we have shown a covalent linkage. Other than that, another set of protein which is present, they are known as a GPI anchored protein, which is present in the membrane, and they are linked to the membrane. And this GPI refers as glycosylated phosphatidyl inositol. We will talk in the detail about this. So this is the complete architecture of the membrane. And here, what you find is that both proteins and lipids, they are present. And there is a small amount of a carbohydrate, which is in the form of either oligosaccharide, which is oligosaccharide present either with the link with the glycoprotein or with the glycolipid. They are the one which are present. So this gives you a overview of membrane, which we have discussed in the previous class. Now, question is whether this membrane is universal. So you have learned is mostly that is a common structure, except. So these variation, let us talk about in terms of their variability in different structures. Normally, what you have seen here is that generally the same kind of structure is present where you have a lipid and you have a protein. Then what you will find is that it may happen in some of the membranes, the sterol portion is not present. Some of the membrane, it may happen that the content of a lipid and protein is variable. So all these things we have discussed. But in general, you will find the same kind of architecture. There is some variation which occurs in case of archaeal membrane. Have you heard of archaea?
Yes, sir.
So they are the one, these are the organism, they are present in very, very, different environment. It is present in a harsh condition. And this is one on the right hand side, the one which I'm showing you with this is a volcano. So here, what you are seeing is that there is a orange color mat. This mat is the one which is the archaeal membrane. So that means they can be present near, these are yellow color sulfur deposits, and on top of it, archaea is present. And this archaea is living in a very, very high temperature. And to survive here, they have certain modification in the structure. Is that their glycerol backbone. So we have talked about the phospholipid. In the phospholipid, you have learned that there is a the glycerol backbone. So you have glycerol backbone having 3 OH group. What you find is that the number 1 and number 2 position, your fatty acid is present. Now, what is the kind of interaction between the fatty acid and the OH group of the glycerol chain? It is going to be ester linkage. So fatty acid COH group and OH group of the glycerol interact, and the kind of interaction you have is ester linkage. But in case of archaeal membrane, what you find is that this kind of bonding is between the fatty acid and glycerol chain, which is a ether linkage. So the linkage differs. So here you have ether linkage in archaeal membrane compared to ester linkage in case of other organisms. Another very peculiar thing you will find that the alkyl chain is a branched one. So you look at any fatty acid, normally what you have is that fatty acid will have straight chain. Here in archaeal membrane, this is the branched chain structure. And third modification which you see in archaeal membrane is that the stereochemistry of central glycerol is inverted. So if I look at this glycerol chain, this is the glycerol one. What you find in the normal, say, animal, plants, and microbial system, that the glycerol, if I number it, suppose this is a syn 1, this is a syn 2, this is a syn 3. So these are the numbers for carbon present in glycerol. So what you find is that generally in all the membranes, the fatty acid is present at syn 1 position and syn 2 position. And in syn 3 position, you have a phosphate group. And then you have phospholipid. Whereas in case of archaeal membrane, this is present at syn 1 position. So phosphate group is present at syn 1 position. And then you have, for example, this is the structure of ethanol amine. So this gives you a very different understanding about the archaeal membrane, where the phospholipid composition or lipid composition is slightly different than the normal membranes. Now, coming to understand that how the sugar unit is attached to the membrane, you have seen in the first slide that glycolipids are present. So these glycolipid, they refer to the presence of sugar unit, which is present in the structure, and this sugar unit could be different. So when I'm talking about the glycolipid, glyco refers to the sugar unit, and this is what has been towards the nomenclature of a glycolipid. So it contains sugar, and one of the simplest glycolipid where a sugar structure is attached to the whole lipid structure is a cerebroside. No need to go into detail of these structure, because this is right now I'm explaining that if I have a fatty acid chain, if a glucose unit or galactose unit is attached with this, we call them as glycolipids. So glycolipids, they are oriented in completely asymmetric fashion with the sugar residue always on the extracellular side of the membrane. So you have to understand this point. This is a very interesting point, that whenever you have a glycolipid, let us go back to this. Whenever you have a glycolipid, this glycolipid, they are present on the outer surface of the membrane. So why should they be present on the outer surface of the membrane? Any one of you? These glycolipids are hydrophilic in nature, and that is why they are present on the outer surface, so that they can interact with the rest portion of which is a hydrophilic in nature. Number one. Number two, I'll tell you how they are, excreted outside the cell membrane. And number three is that they are the one which are the structure which maintains the asymmetry of the membrane, which we talked in the last class. Asymmetry means if I break down this membrane into two halves, I'm just breaking it down in two halves. So here, the lipid structure in this half will be different than the lipid structure in this half. Of course, I have not shown the three dimension on the back side, but the similar structure is present. Now, what will be different is that the phospholipid present here may be different. The phospholipid present on this side will be different. So these two are asymmetrical. Same way, the protein structure present over here may be different from the outer side and the inner side. Okay, now let us talk about the cholesterol, which we have talked in the membrane structure. It is a sterol group. It has a very different structure than the phospholipid structure, but still it is a part of the membrane. Why it is a part of the membrane? Because if I look at this structure and if I look at the general principle of the membrane structure, any lipid which has got both hydrophobic and hydrophilic group can become part of the membrane. And if I look at the cholesterol, in the cholesterol, can you see both hydrophobic and hydrophilic part? So you have a huge hydrophobic part. So this part is a huge hydrophobic part, and there is small tiny portion which is a hydrophilic in nature. Can you see?
Yes, sir.
Okay. So very small portion of the hydrophilic OH group is present here and rest is hydrophobic. Now this steroid is built up of 4 linked hydrocarbon chains. So if I want to number, you have first, second, and third, and this is the fourth. These 4 rings are fused. Can you name this ring? I think I am just circling first 3 rings. Can you recall this ring? The first 2 you have learned in organic chemistry, first 3 also you have learned. It is a phenanthrene ring. First 2 if I take, it is a naphthalene, and the first 3 if I take, it is a phenanthrene ring. So if I just give the architecture of this, I can say that the cholesterol, the basic ring structure is phenanthrene here, and there is a cyclopentane. So there is a pentane which has been cyclized. So you have parahydrocyclopentano-phenanthrene ring. It's a big name, but it just gives you that the basic structure is based on phenanthrene ring, where a pentane, cyclic pentane is attached, and then you have another chain which is associated with it. So what do you expect from the cholesterol? Cholesterol has to provide membrane lot of hydrophobic nature of the ring, isn't it? Huge portion of this cholesterol is hydrophobic.
So,
So
question.
yes, any
So,
question?
why do we consider this as a phenanthrene ring? Because it doesn't have a benzenoid structure.
I have not shown it to you, but this ring structure, there is a modification. So if I look at the benzene ring, so whenever you have a phenanthrene ring, you have 1, 2, 3, and then you have a double bond. Now
Yeah.
this double bond has been satisfied in many cases, but it gives you the basic unit structure of phenanthrene ring. Of course, at this position, 5, 6 position, there is an unsaturation. Third position, your hydroxyl group is attached. If I look at the core structure of the ring, it is a phenanthrene ring. And that is why it is called as perhydrocyclopentano-phenanthrene ring. So that is the basic unit from where cholesterol has been derived. Now, of course, there are certain positions where your saturation has occurred. Okay. I'm not going to debate on the organic chemistry part of it, but the basic point is that it is giving you a lot of hydrophobicity in the structure. Then, in case of a membrane, approximately it may constitute around 25% of the membrane lipid, and in case of nerve cell, but essentially it is absent in the intracellular membrane. So suppose I have mitochondrial membrane or some other membrane, it may be absent. Now, looking at uh, the one where they are present, let us go back to the first one. So where do you find this cholesterol present? This cholesterol is present in the fatty acid chain where they are present in the membrane. It could be on the inner surface, it could be on the outer surface. So both sides, this cholesterol is present, and it's most of the interaction is with the lipid. Now, the question for you is that where should you expect OH group?
So on the outside also.
Yeah.
So
So OH group will be present either here. Here OH group is protruding outside, or OH group will be protruding on the inner surface on this side. Rest part is a hydrophobic. So it can have very good interaction with the membrane lipid, which is a phospholipid fatty acid chain. So I think so far it is about the membrane fatty acid and lipid composition. Now, this lipid about, I'm explaining, elaborate it a bit more. The membrane lipid is amphipathic molecule. As I showed you in the previous slide, that cholesterol is amphipathic. That means both hydrophilic and hydrophobic part. Here also, most of the membrane lipid, which we have talked about, they are phosphoglyceride. They have both hydrophilic and hydrophobic part, same way sphingomyelin. And even in archaeal lipid, you have both hydrophilic and hydrophobic part. It is important that hydrophilic unit is also called as a polar head group. So if I want to give a shorthand depiction of this is the hydrophilic part of the membrane, and this is the fatty acid chain. It is always shown as a wavy structure. So whenever we have seen it in the membrane, this structure this structure represents the phospholipid. And the chain, these chains are fatty acid chain. This part is showing the alcoholic group of the phospholipid. Now, this also gives you some more idea that phospholipids and glycolipid. Why glycolipid can be form of the membrane structure? Because why should glycolipid will be part of the membrane structure? Which one is a hydrophilic in glycolipids? The glyco portion. So the glycose, which is a carbohydrate, it has got several OH group, and that's why they will be hydrophilic in nature. So if I take phospholipid and glycolipid, they tend to form the molecular sheet-like structure. And what molecular sheet you have is the membrane, is a biological membrane. So bimolecular sheet-like structure is a form if I take phospholipids or glycolipid in water or in any other aqueous medium. So membrane formation is a consequence of amphipathic nature of the molecule. So if I look at the membrane structure, we can say that if I have phospholipid, which is both hydrophilic and hydrophobic, they will part a membrane structure. So it is a self-assembly which occurs. So if you recall our first and second lecture, where we have talked about the self-assembly, where we talked about that how the interaction between non-covalent interaction occurs between different molecules. This gives a unique example. So what happens? You have a hydrophilic part, you have a hydrophobic part, and membrane lipid contains both. And what happens is that it can satisfy a bilayer type of structure. And this bilayer type of structure is formed in order to expose them, their hydrophilic part, to the hydrophilic part of the inner side of the membrane and hydrophilic part of the outer side of the membrane. In between, it is a hydrophobic fatty acid chains. So these hydrophobic tails of individual sheets. So if I take two sheets and there is a hydrophilic part completely, maybe outer surface of the membrane. There is again hydrophilic part on the inner surface of the membrane, and in between you have a fatty acid which is hydrophobic in nature. So this is the hydrophobic part of the membrane lipid which acts as a permeability barrier. Now, the favored structure of the most phospholipid and glycolipid in aqueous is a bimolecular sheet-like structure. Why bimolecular sheet-like structure? Because if it is an open chain, suppose I have just half layer, one molecular layer sheet. So what will happen? This fatty acid chain will be exposed to the water and that will be unstable. They will try to aggregate, they will try to fold back. So this kind of structure is not present. So what is the structure of the membrane lipid? They are bimolecular sheet, and each sheet will have hydrophilic and hydrophobic part. So that's the way they are organized. Now coming further to this, the favored structure is bimolecular sheet rather than a micelle. Now what will happen? In the micellar structure, if I create a micelle, it will be a very small portion. So the membrane, which is present, for example, outside the cell, it needs a longer structure. So it's a basically steric thing which controls the formation of a bimolecular sheet. Some of you may argue that why not a open sheet? Because it is organizing in the cell, it can become as an open sheet. But tell me what will be the problem if I have a self-assembled membrane structure, which is an open sheet structure? Any one of you?
So what do you mean exactly by open sheet?
Open sheet is that I'm not going to close it like membrane. It is present, membrane is a present just like a sheet-like structure, and all the assembly is associated with the membrane. You in the case of mitochondrial membrane, a lot of enzymes they are associated. So what will be the issue here? The issue is, major issue, if you look at, if I leave these two sides open, the water will start interacting with the hydrophobic chain, and that is a non-stable structure, and that's why together. For example, this membrane and this membrane, they will close, and that's the way they can protect themselves from water. Is it clear to you?
Yes, sir.
Okay. So now we move on to that formation of a lipid bilayer is a self-assembly process. Self-assembly means it does not require any energetics. It does not require extra ATP to synthesize it. Whenever you put them in the water, like you have seen in the micelle formation, they will start making a sheet-like structure. So the driving force, the hydrophobic interaction, or the major driving force for the formation of lipid. So there will be a lot of interaction between the hydrophobic fatty acid chain, and they will, what kind of interaction they will have? They will have a van der Waal interaction. Because of this van der Waal interaction, they will start making a bimolecular sheet. Now, another kind of interaction that occurs with the phospholipid head group and the water molecule. So what will happen, that water molecule, they will get associated with this, and there will be cooperative interaction, and they will form a complete structure. And why water can interact with this? Because water is hydrophilic in nature. They will be having certain charge, and phospholipid group will also have a charge, and that's the way they can interact. Now, coming to the question, if it is so, and if I take the membrane, take out the lipid from a plasma membrane of any organism and put them in water, can we form a structure? Answer is yes. You can form lipid vesicle. So to form a lipid vesicle, what you require is you require lipid, suitable lipid. For example, you can take phosphatidylcholine, or you can take phosphatidylethylamine, anything which is having both hydrophilic and hydrophobic group. So lipid vesicle are liposomes. They can form aqueous compartment, and they are enclosed by a lipid bilayer, and they can be, so the question comes where you are going to use. They can be used to study the membrane permeability as well as to deliver chemical. Here, when I'm talking about the chemical, it could be the drug molecule. So if I encapsulate drug over here, it can be delivered in the cell, and it will interact with the cell. There will be fusion of this membrane with the original cell membrane, and that's the way the drug can be delivered inside the cell. So the liposomes, they are formed by suspending the suitable lipid, such as a phosphatidylcholine, in aqueous media, and then you just sonicate them, they disperse them, and you will get these closed vesicle. These are more or less typically the same structure which we have talked about, bimolecular sheet, but then they are circular shape. If I look at why it is not formed in case of cell membrane, because the kind of dynamics they have here is that the inner core is a very small. So this kind of a structure may not exist unless we make sheet like structure where the structure organization is much bigger. So another well-defined synthetic membrane could be planar bilayer membrane. So instead of a circular layer, you can have a planar layer. And this planar layer can be formed. You can synthesize these planar bilayer membrane structure by using phosphatidylcholine, creating 1 millimeter hole in partitioning between two aqueous compartments. So these are the details given for their formation. You have the decane, and on that you are putting phosphatidylcholine, and then you use the paintbrush, and that's the way this is being formed. This is the complicated structure. We are not going to focus on that, but yes, the formation of liposome is a very common, and you can make it in the lab. It's a very simple procedure. You take the lipid, put them in a buffer, remove the, first you take the lipid, putting in the hexane or in the chloroform, remove all the solvent, then put them in a buffer, sonicate it, your membranes are ready. Now, if I look at the permeability, what kind of a permeability of these membranes they have? So lipid bilayer membrane have a very low permeability for ions and most polar molecule. So suppose I make a animal cell or bacterial cell membrane, which is just made up of lipid. What is your about the permeability? In natural form, they have a proteins. Now I make a lipid bilayer which is lacking any protein in the structure. What will happen? If they do not have any, there should be zero permeability. So water, except for the water is also hydrophilic, but they are freely permeable across the membrane. But any other molecule which is, hydrophilic, they will not be permeable. And this is because the water has a exception, and It can traverse such a membrane because of the small size, number one. But size is not the only thing. You have a high concentration and the lack of complete charge. So there is a not. So if I talk about the water, you always say oxygen is having partial negative charge and hydrogen is a partial positive charge. So these are partial charges. These are not complete charges, and that's why probably that becomes permeable. Looking at their permeability behavior, you can see that smaller molecule like sodium ion and potassium ion, they are also highly, restricted for being transported across the membrane. We will talk about some of the example where these molecules, they are transported across the membrane with the help of proteins. So the proteins which are present in the membrane, they are the one which is a functional molecule, whereas a lipid provides the barrier. Then you have a glucose, you have a tryptophan. So I have given you just kind of understanding that these are the molecules depending on their hydrophilic nature as well as the size, their permeability varies. So you find the increasing permeability here. Water is a highly permeable and sodium ion is the having lowest permeability. So it is based on the charge, it is based on their structure as well as some of the interaction with the membrane. So when I'm talking about this kind of a permeability, it is based on their membrane lipid composition. If the membrane lipid has got a specific transporter, for example, if I have a sodium transporter, it will transport sodium ion. If I have a glucose transporter, it will transport glucose ion. So in the membrane, for transporting the hydrophilic molecule, you will have several transport. So let us talk about, so far we have talked about the structure of the lipid, we have function of the lipid. Now let us talk about the function and the structure of the proteins. Now these proteins which are present in the membrane, they are the one which are, we will talk about the composition. So let's first talk about the membrane lipid are the one which are forming the permeability barrier. And thereby they establish a compartment, whereas specific proteins which are present, they mediate nearly all the membrane function. So most of the membrane function is performed by the proteins. So they are the one which are actors. And what lipid does then? So the other day we were talking about the lipids. What is the role of lipid? The lipid provides them the environment where such interaction can occur. Somebody asked in the last class that if they are a fluid, is it not possible that the proteins they will interact? Answer is yes. If they're fluid, the protein can interact with each other. Lipid can allow them to be fluid. So those portion we are going to discuss further. So membranes differ in their protein content. It means that the one membrane can have a different protein content than the other membrane. Though they will have both of them, they will have a protein, but the example, if I have to transport through a membrane sodium ion, I will have the transporter for sodium ion. If I need to transport glucose, I will have a transporter for glucose. For example, mitochondrial membrane will have a huge number of enzymes which are responsible for the energy production or ATP synthesis. So that way, each membrane will have a different composition. So I have taken example, this we talked in the last class also, that myelin, a membrane, where does this myelin is present? In the nerves.
Nerve.
Yeah. So it is a membrane that serves as an insulator around the nerve fiber. So around the nerve fiber it is present and it is acting as an insulator. So what is expected here is that it will have very high content of lipid, because lipid is the one which acts as an insulator molecule or permeability barrier molecule. And that's why the myelin has got very low content of a protein. So that is where I have talked about composition varies from 1:4 to 4:1 between lipid and protein. Now, relatively pure lipids are well suited for insulation, whereas a plasma membrane or exterior other cells are much more active. So what it tells you is that if I just take a pure lipid, it becomes completely insulator. So what you have is that you don't have a completely insulating membrane, rather you have a membrane which has got lot of other protein for different functions. And these functions can be performed by pumps. Pumps means the one which can allow the molecule to move in. It should have a channels. I'll tell you what are the channels. Channels and pumps they are more or less of same kind of a function, but by a different mechanism. That means both of them, they will be allowing the molecules to move across the membrane. Then you have a receptor which can allow the binding of the ligand molecule to the membrane. Then you have enzyme, as I discussed in case of mitochondrial membrane, you will have several enzymes which are performing function. Then protein content of these plasma membrane are typically 50%. So I'm talking about the plasma membrane, which has both insulator function as well as a function like a pump channel receptor enzymes. So they will have a 50% enzymes, 50% proteins and 50% lipids. Proteins could be variable, as I said, pumps, channels, receptors, and enzymes. Now, coming to the energy transducing membranes, as I have discussed that the mitochondrial membrane or chloroplast membrane, they have energy transduction function, where you will find that a lot of the membrane is made up of protein. So that function is to perform some reaction, so there is a lot of enzyme which is present in mitochondria to perform such functions. Now, plasma membrane of erythrocyte, photoreceptor membrane, or retinal rod and other membrane of muscle contain many proteins, but has a distinct protein composition. So what I'm trying to say here is, suppose we have a retinal cell, we need to transfer the photons which are interacting with our retinal cell. Where is the retina?
Eyes.
Eyes. So you have a retinal cell which is performing the acting as a photoreceptor. They will have a different protein composition than the sarcoplasmic reticulum, which is present in the muscle. So each one will have a different composition of the protein. So protein is the one which is giving you characteristic function of the membrane of any cellular organelle or cellular structure. So that is what, in general, membranes performing different function will have different repertoire of proteins. Now, looking at the further proteins, though I have discussed in general, but let's talk about this. So protein carry out most of the membrane processes, and membrane protein can be classified into three different types. I have shown, I think, two of them here. One more I will discuss at the later stage. One is the integral membrane protein. This is the one yellow color, which is across the membrane. These are the one which is interacting with the hydrocarbon chain of a membrane lipid. So only agents that compete for the non-polar interaction can release them. So suppose I have some reagent which can interact with a non-polar environment. So which reagent over here? Mostly the organic solvent. They can dissolve the lipid, and these integral protein can be liberated. In fact, most of the integral protein span the lipid bilayer. So they are spanning the lipid bilayer across the membrane they are present. Looking at the peripheral membrane, for example, this blue color or this blue color, bound to the membrane primarily by electrostatic or hydrogen bonding with the head group of the lipid. So lipid head group, they can interact with these outer side protein, or it can also interact with the surface of integral protein. So they can be present. This is integral protein, and the surface of integral protein, this is present. So it could be a protein-protein interaction. It could be protein and lipid interaction. So that is why they are interacting. The third type you will find anchoring to the lipid of protein. So let us talk about that. I think I will discuss that little later. So these are the some protein, just to give you idea that there could be protein which spans the membrane with alpha helix. So these are alpha helical structure. These alpha helical shown. The channel protein can be formed from beta strand. So channel protein, you will see that they are the beta strand. So these are the beta strand which are present. They are embedded in a membrane and can link. So you can have embedding part of the protein in the membrane, which can link it to the membrane surface. So it is linked with the membrane surface. These are just depiction of different types of the membrane, which is linked with the proteins and showing how they are interacting with the rest part of the lipid. Now, the third part, where I have discussed the sum of the protein, they can be associated with the membrane through covalently attached hydrophobic group. So these are the one which we have shown you. So they are the one, they are associated with the membrane through the surface generated by hydrophobic amino acid in the side chain. So you have a side chain hydrophobic. They are allowed in the binding. Now, soluble protein can also be associated with the membrane if the association is mediated by the hydrophobic group to attach with the protein. So there are three variations which occurs. One is where a palmitoyl group attached to the specific cysteine residue by thioester bond. So this is a thioester bond which is present between the protein structure and rest part of the membrane. Then another fatty acid, pherencyl group, they are attached with the carboxy terminal of cysteine. So you are seeing that it is interacting with the cysteine residue of, where is the cysteine present? Cysteine should be present in proteins, and they are interacting with the either palmitoyl group or pherencyl group. So these are fatty acids. A glycolipid structure termed as a glycosyl phosphatidyl inositol or GPI anchor is attached to the carboxy terminal. So these are the structures which are given, which shows that how they are anchored with the membrane protein. So far, what is the difference here? Here the difference is that generally the protein and lipid or protein and protein interaction which is occurring, they are non-covalent interaction. Here we have given the example where the covalent interaction allows the lipid and protein interaction. Am I clear?
Yes,
Have
sir.
you understood this concept? So let
Yes.
me just give you the very basic, the first slide, where I have shown you this GPI anchored protein, the top one. Can you see that? It is linked by how? Through the, it is linked with the, this is the protein which is anchored, and it is linked with the membrane by covalent interaction. Whereas if I look at this lipid and integral protein, their interaction with non-covalent interaction. The hydrophobic groups of the protein and the hydrophobic fatty acid, they will interact together. But here what you find is that this interaction is through covalent, whereas this interaction is non-covalent interaction. So if there is a non-covalent interaction, they will be removed by simple, sonication or some physical means. But if there is a non-covalent interaction, you require more energy or you need to break the bonds. Okay, just quickly we will discuss one or two more slides and then we will stop. Now, if I say that if I have looked at the structure, I'm just going to some slides away. So this is the structure which is very well established, which shows that membrane exists as a fluid and the proteins, they are present as a mosaic, and lipid is the one which is making the movement, or the lipid is the one which is making a fluid like structure, and proteins, they are present as an iceberg. So it's a classical model, Singer-Nicolson model, which has been given. So what it explains is that in this fluid structure, so a little bit, that if I take the lipid portion over here, that means these lipids which are present in outer layer, suppose I take this as an outer layer. So any lipid or protein, they are movable over here. So suppose lipid is present here, it will move to this portion. So from second, it became third. So this is a kind of a diffusion which is called as a lateral diffusion. Whereas there is another type of diffusion which may occur. That means lipid from here moves to the other layer. So it moves from one layer to another layer. So this is called as a transverse diffusion. So these two diffusion may happen. But generally what will happen, the diffusion from one layer to another layer, that means trans bilayer movement, will be restricted. And why should it be restricted? As I said, that this portion is a hydrophilic. So if a hydrophilic molecule goes through the hydrophobic portion, there will be energy barrier. And unless there is an enzyme or unless there is some support, they will not move across the membrane. So in some viruses, there are enzymes which allow this transverse movement, but in general, in most of the proteins, such movements are not possible. So this gives you an idea that membrane shows the movement. So here, the fluid mosaic model allows the lateral movement, but not rotation through the membrane. So on the basis of the dynamics and properties of the membrane, Singer and Nicolson has given the membrane model where it shows that the membrane is a two-dimensional solution of oriented lipids and proteins. It is the same thing, that it is like a sea where around in the membrane. And here, what he called is that transition of a molecule from one surface of the membrane to another is called as a transverse or diffusion of or flip-flop. So if you call it as a transverse diffusion or a flip-flop movement, and the flip-flop of phospholipid molecule in phosphatidylcholine vesicle, he has done experiment and he has demonstrated that if I take phosphatidylcholine and look at the flip-flop movement, it is not occurring, or it is occurring in several R, one molecule transfers from one layer to another layer. In fact, this flip-flop movement of protein molecule has not been observed by him, and hence membrane asymmetry can be maintained. And this is because of the free energy barrier which is present because of the presence of a hydrophobic molecule or hydrophobic structure. Now question comes, can we demonstrate the same thing over here? First thing is that how to demonstrate that membranes are a fluid structure. So that part has been demonstrated by doing a very simple experiment. It is a fluorescence based experiment, and it is called as a fluorescence recovery after photo bleaching, or it is termed as a FRAP technique. So this FRAP technique is a very simple technique where you can find out in a given time period how much membrane is moving from one place to another place. So we'll go into detail, and it's a very simple technique by which you can take a cell, you can take a membrane, you can label this. So suppose this is the membrane. If I see under the fluorescence microscope, it will look like this. And then what you do is that you pass a laser light in a small portion, this fluorescent will disappear. So this is what you see. And if I leave for certain time, if membrane is a fluid, what will happen? These molecule, which is a fluorescent molecule, they will come and recover this place. So whatever you have created a bleach can be recovered. So this experiment I will explain you in a greater detail, and will give you the significance that you can find out the average distance, that is S, traversed by any lipid molecule or a protein molecule in a given time. So with this I'm stopping. I will continue with the FRAP technique, give you detail how you can use it and what are the possible movement of the different molecule in the membrane lipid. So tell me if you have any quick question. Otherwise, we can discuss the question in the next class. Any one of you?
So I had a question regarding the archaeal membrane. So, like as we stated, it has a one phosphoglycerol ether linkage instead of three phosphoglycerol ester.
Right.
So, what advantage does it afford to archaea, like depending on the environment it
So
is
they
in?
become more stable. So it is the type of a bond present, and not only that, there are other modification also occurs. But these are the adaptive mechanism where lipid has been made more stable compared to the lipid which is present in the normal bacterial membrane or animal cell membrane. Slowly the looking at the stability, bacteria, they have a cell wall because they need, harsher environment. Animal cell doesn't have the cell wall. You have a plant cell which has a cell wall, but animal cell doesn't have cell wall. So these are the something which has been adapted, something which has been dropped down. So unless there is utility of some part, it is not being accepted in the evolution portion. And that is where you find that the bacterial membrane, which is stable in a harsh environment, archaebacteria, which has a harsher environment. So that is where you find their genetic material, their membrane, all those things, they have a unique structure. So that provides stability to the membrane. Anything
Oh,
else?
thank you,
Any other question? Any one of you? Okay, so if there is no question, I'm going to sign off. Thank you very much.
sir. Thank you, sir.